Hello, my name is Jina Kwon, and I will be presenting about seagrass transplants and effective transplant methods to accelerate seagrass recovery. To begin, seagrasses is an organism that is in most times given less attention to than other marine animals or plants. During a trip to Belize in the summer of 2019, I was first introduced to the important value of seagrasses. This inspired me to do further research. Today, my presentation will consist of an evaluation of different methods for seagrass recovery and a proposal to combine several existing transplanting methods using developing technologies for a more effective transplant system, with a focus on accelerated seagrass recovery. Before starting, I would like to take a minute to provide an introduction of the background information necessary to understand the later parts of this presentation, as seagrasses may be an unfamiliar topic to many. Seagrasses are categorized in a group called angiosperms. Angiosperms are flowering plants, but unlike some other flowering plants, seagrasses can live in saltwater environments. They can be found in shallow, salty, and brackish waters in many parts of the world, from the tropics to the Arctic Circle. Often unnoticed and undervalued by many, seagrasses surprisingly play a crucial role not only in the marine environment but also environments outside. For example, many organisms rely on seagrass as a habitat and food source, including some endangered species such as sea turtles, dugong, manatee, and sea urchins. They also play a major role in controlling sediment levels in the sea and controlling erosion, managing appropriate wave velocities so that the organisms can live in. This is important because different organisms can only live in certain wave velocity ranges. Therefore, the biodiversity of the organisms would be highly affected by how fast or slow the wave velocities are. Also, seagrasses along with other marine plants are responsible for 45% of the photosynthesis that occurs on Earth. Even among marine plants, seagrasses are an impressive photosynthesizer with the highest carbon sink. Seagrasses also have economic effects in its surrounding communities. According to a study done by University of Washington, the loss of seagrass is leading to approximately 3,500 hectares per year, and if restored, saves 1.9 trillion per year in the form of nutrient cycling. The loss of seagrasses also affecting the fisheries can lead to negative economic consequences for the fishing industry and some members of the community who might rely on fishing as a living. The current status of the seagrass population is decreasing at a rapid rate. According to a study done by the School of Marine and Tropical Biology, seagrasses have been decreasing at a rate of 110 kilometers squared per year since 1980, while the rate of decline has been accelerating from a median of 0.9% per year to 7% since 1990. In response to such rapid rates, many transplanting methods, such as seeding methods, methods using root stimulants, and other vegetative transplanting methods, have been used. However, results of previous studies have led to controversial debates, as some results show high success rates while others show almost 100% mortality rates. Moreover, though the transplanting was successful in most cases, the restoration and recovery of these beds in the long term have been difficult to remain. With these contrasting results and limitations, solutions to the seagrass recovery have been difficult to conceptualize. Now for the global threats. There are three main global threats that cause seagrass population to decline and these are anthropogenic impacts, coastal development, and lack of recognition for its value. Anthropogenic impacts or human impact include activities such as increasing turbidity, physically damaging the seagrasses, or aquaculture development causing harm in seagrass communities. Loss of seagrass is also highly linked to coastal developments as an onset of land reclamation, land management, or other activities. Along with these threats, the lack of recognition of these benefits and value of seagrasses when compared to other marine ecosystems lead to lack of research and understanding on how such activities can be a threat to the community. Next are the methods of seagrass population recovery used right now. The current methods that are being used right now are mostly natural recolonization, transplants, and seeding methods. Natural recolonization is a method modifying the environment of the seagrass beds to encourage natural recovery. Transplanting methods use transplant donors from other or nearby areas and plant them in the areas where the grass has disappeared. Seeding methods use seeds of the seagrasses to plant in the areas. Although seeding and natural recolonization methods encourage more natural methods, the risk of failure are higher and the process is very slow. Transplanting methods, meanwhile, are very fast processes, which means it can accelerate the rate of global seagrass recovery. Transplants also mean more control over the environment, meaning lower risk of failure. To face the quick rate of declining seagrasses, I had chosen to narrow down my topic to transplanting methods, as I thought that it was quick and effective, making it the most appropriate method. So the two questions foundational to my research are: first, what are the possible natural and technological solutions for the global seagrass loss? And second, what are the effects and limitations of seagrass transplanting? Specifically, here, natural solutions refer to the changing the environments of seagrasses so that the seagrass will be in an environment where they can naturally recover on its own. Technological solutions refer to using technology to help with the process through the recovery. Both my questions were aimed to help myself navigate into identifying the current limitations and way to solve them. Formulated around those two research questions, my thesis statement claims that seagrasses are essential in maintaining the biodiversity in marine ecosystems, and through improved modern transplant technology, regions with problems related to seagrass loss can be solved. Here, the problems refer to a wide variety of negative influences that seagrass loss can have, including decreased diversity due to lack of food or shelter, strong wave velocity that is caused by the absence of seagrass. Next, I'll be moving on to explaining some terms that will appear in the next couple of slides of my literature review. Eutrophication is a process by which a body of water becomes over-enriched and dissolved in nutrients that stimulate the growth of aquatic plant life, usually resulting in the depletion of dissolved region. Epiphytes are organisms that grow on the surface of a plant and derive its moisture and nutrients from the air, rainwater, and marine environments, or from the debris accumulating around it. Bistability is the phenomenon in which a dynamic system can exist in one or two stable states for a given external condition. Now I will be moving on to the literature reviews. The purpose of this literature review was to compare different current challenges of transplants and restoration of seagrass meadows with the emerging technologies that show potential in solving such limitations. Studies of varying transplant methods that have been applied to different environments were compared separately. First off, the Marine Ecotechnology Institute conducted a study called A New and Effective Transplanting Technique Using Wired Netting for Restoration of the Surf Grass, Philospadics japonicus, in exposed rocky shores. This study focused on evaluating the performances of a new method, utilizing a sturdy stainless steel wire rope and affixing the transplants of surf grass, a type of seagrass. Seagrasses play a big role in regulating the wave velocity, making sure that the shores are not too rapid. In areas going through severe seagrass loss, the turbulence increases accordingly, and this can become a hurdle in the restoration processes, as the turbulence prevents the growth of newly transplanted seagrasses. This study has a geographic focus on the rocky shore of South Korea, and most seagrass transplant methods have been focused on shallow, slow wave waters, making the methods made prior not applicable to areas with more rapid currents or with greater depth. The experimental group that had been affixed with the wire netting method had shown that the transplants rapidly increased at first, but as the rate of growth decelerated, the growth rates ultimately reached similar size and growth rates as the pre-existing seagrasses. And this study focuses on using developing technology to fix the limitations, and thus it was one of the technical methods that I found in my research. Another study that I referred to was determining the effects of root cell manipulation on seagrass to enhance restoration projects by Eric Milburn, along with members of the Marine Laboratory, Sanibel Captiva Conservation found in 2007. This study primarily focuses on how different levels of sulfide, either plus or minus, and how the presence of autoclave can affect the mortality rates of root transplants and plug transplants. The bare root transplants had a significantly low rate of growth compared to the rate of the growth of the plug transplants, suggesting sensitivity of root disturbance in seagrass transplants. The seagrass growth rates were also significantly lower in autoclave sediments than in native sediments, suggesting the importance of bacterial communities for restoration success. This study focuses more on environmental modifications, making it one of the natural solutions in my research. Next, feedback between sediment and light for where is it important, conducted by members of associations of science in limnology and oceanography, was also one of the studies that heavily influenced my conclusion of this research. This journal also would be in the natural waste categories. It identifies how the environmental and seagrass meadow characteristics influence the strength and direction of seagrass sediment light feedback, which will be called SSL feedback in this journal, and how this feedback had been incorporated in the ecosystem models proposed to support environmental decision making. The conclusions made by the study reveals that the feedback that can be used to identify where the bistability occurs, allowing separate treatment to be done in these areas. The study done in 2010 by A. Randall and members of the Coastal and Marine Laboratory, Florida State University, called Genotypic Diversity and Grazer Identity Interactively Influence Seagrass and Grazer Biomass, emphasizes the effects of seagrass genotypic diversity and grazer species composition on seagrass recovery. Using the species Costera marina, along with diverse grazer species and epiphytes, the study shows that seagrass genotypic diversity does not affect the reproduction of seagrasses, but only the plant itself and the grazer species, as they act as a food and habitats for the grazers. However, the existence of grazer species show larger effect on the growth rates, thus coming to a conclusion that during the recovery period, the biomass of seagrass is significantly higher in polyculture than in monocultures. Next, the 2011 journal Seagrasses and Eutrophication Interactions Between Seagrass Photosynthesis, Epiphytes, Macroalgae, and Mussels by Esther Frances Mavungi explores the interactions of seagrasses with increases in nutrients, ephemeral age, and changes in the physical chemical properties of seawater induced by interaction with other marine biota. The increased concentrations of water column nutrients had stimulated growth of the epiphytic biomass on seagrass leaves, thus negatively affecting the seagrass photosynthesis. However, the interactions with certain organisms and the surrounding environments, including mussels, Pinna muricata, and seagrass type Thalassia hamprichi, enhanced the photosynthetic activity of the seagrass, while there was no evidence of any calcification of the mussels. Lastly, integrated planning that safeguards ecosystems and balances multiple objectives in coastal Belize was a journal that had both inspired me to start the research and also ended with further suggestions. It includes explanations on some of the organisms highly dependent on seagrass and are becoming extinct partly due to the declination. I thought the application of the conclusion, or seagrass restoration in general, is very relevant to Belize. Now, before moving on to the results, I would like to explain up some terms I will be using in the next few slides in order to prevent confusions or misunderstandings of the results. The p-value, also called statistical significance, often expressed by numbers between 0 and 1, measuring the level of significance, will be used in the next slides. The smaller the number, the stronger the significance. Normally, studies with p-values under 0.5 are viewed as significant. Next, the mortality rates that are going to be in the next slides will refer to the rate of death or failure of the transplant after the transplant has been done. Survival rates will also be the likeliness of regular growth after the transplant process. Now I will be explaining the results of some of the key researches discussed in the literature review. Among the many journals exploring different ways to change the transplanting process, the following 4 studies of the results of the studies are the ones that I had concluded to be the most effective and will act as the foundation of the conclusions made in the end. So this is the results of a study regarding sediment and light importance in the biostability of seagrass beds. Figure 1 shows the cycle in which the seagrass is lost. The simultaneous loss of ecosystem engineering benefits may hinder its recovery. In other words, the turbidity of the water and wave velocity will increase with the loss of seagrass. These factors build up the SSL feedback, which was found to be a major barrier to seagrass restoration. Likewise, studies in Dutch Wadden Sea show that the significant seagrass loss had not been able to be reversed despite great management of the surrounding environment and extra research. However, other areas that have been studied, such as Virginia Coastal Reserve, have shown successful restoration despite the increased turbulence. This proves that increased turbulence is only one of the many factors for seagrass meadows' inability to recover. Additional research in this journal that in some location in the SSL feedback may cause by stability, the seagrasses are more resistant to loss when present, but more difficult to restore once lost. One of the 7 linked processes, suspended sediment linked to attenuated light, shows a greater significance in the results. Seagrasses require photosynthetically active radiation with wavelengths varying from 400 nanometers to 700 nanometers. 11% of the surface light extends to the seagrasses. In Figure 2 demonstrates the SSL feedback on the lower potential and higher potential systems and the environmental impacts of 5 seagrass meadow characteristics, which are meadow depth, eutrophication, meadow size, sediment size, water velocity, can have on the feedback loop. The results demonstrate that for lower potential areas, the meadow depth is deep, subtidal, has high eutrophication, sparse size, narrow range of sediment sizes, and lower water velocity. These impacts come together to show that the SSL feedback was unlikely to make seagrass decline difficult to reverse, and that seagrass restoration in such areas have possibility to occur naturally, whereas higher potential areas has a low possibility of natural restoration and seagrass loss. The next results are from the study regarding genotypic diversity and grazier identity on seagrass biomass. Figure 1 shows the growth of the transplant Philospadics japonicus, which was the experiment seagrass used in the study. There was significant change in the relative growth rate of the transplanted Philospadics japonicus with a significant difference below 0.001, below the significance difference for which studies would need further research. Leaf growth was initially higher in transplanted areas than the Natira population for 15 months, as was the leaf growth rates was. After the rapid growth for the 15 month, the growth rate decreased and the levels of transplanted seagrasses leveled to be the same as the natural population, thus preventing further eutrophication. Figure 2 shows the contrasting changes of the seagrass populations in the transplanted areas, above ground and below ground. During the 23 months of the observation period, the growth rate of the transplanted Philospadics japonicus reached 144.4 millimeters and displayed 41.7 nodes, showing that transplanted rhizomes growth rates were 66.7 millimeters per year. Their weight gain rate was at 203.5 milligrams per year, while the average interval was 19.8 days in the transplanted Philospadics japonicus. The fourth results are from the study exploring the effects of the different NAPH, naphthalene, acetic acid, and substrate types on biomass. The study demonstrates the importance of the adequate concentrations of the NAPH and appropriate amounts of substrates. Figure 1 here shows the example model used in the journal of the aquaria containing the transplants. The model was built to make sure that there is a constant flow of seawater and sediments in the transplant's aquaria. Figure 2 shows the survival rates of the control groups and the groups that were treated with different concentrations of NAPH. Haduliridi, which is the seagrass used in the study, was found to be only surviving in all NAPH-treated tanks, but only 2 in the control groups. Survival rates were also highest in transplants treated with 0.5% to 1%, which were the concentration rates of root dip, which was a commercial brand root hormone used in the study. For the first months after the transplants, 87% of the treated groups had survived compared to 38 to 39% survival rates of the control groups. After 2 months, the survival rates had dropped out but were still significantly higher than the control groups. The declines in survival rates may be explained by epiphytic growth, which was somewhat a problem during the observation period of the study. Lastly, the results of the study, a new and effective transplanting technique using wire netting for restoration of the surfgrass Philosphatus japonicus in exposed rocky shores, done in 2018 by Jungin Park and members of the Korean Marine Ecotechnology Institute Busan, also shows a different approach to solving with problems in transplanting methods. This study, being a relatively new one, shows positive results, especially on the application of transplanting seagrass beds in rocky areas. In Figure 1, the sea growth ground weight on the y-axis is shown to increase according to observation time on the x-axis. The leaf growth and height had significantly increased faster in transplant groups using the wire nettings than the control group. Similarly, Figure 2 shows the overall growth through the change in length, weight, and number of nodes. The transplant's rhizome's growth rate was 66.7 millimeters per year, which is a very fast rate, but gradually slowed down to 19.7, ultimately letting the seagrasses level to the rate of the original seagrasses. All in all, the wire netting method showed to be successful in the long term without reaching overgrowth, which might lead to eutrophication. This study also relates to the results of the study regarding the SSL feedbacks, as the netting can help solve the limitations of the turbulence interference mentioned in that journal. Now, based on the results, these are some limitations that are byproducts of the results or elements that limit the effectiveness of the results. First of all, one of the limitations would be need for further research. Although the individual limits and effects can be explained, once combined, there might be other factors that might alter the results. Another crucial limitation will be human interaction. Although controlling environmental factors can be done, there is no way to restrict human activity with these methods. Human activity has the greatest impact on the seagrass loss, especially through eutrophication. In this case, human impacts may interfere with the process and change results. The photo on the left shows the image of site after eutrophication, and the photo on the right above shows how human activity causes eutrophication. Moreover, the limit of financial support would hinder the process of further research and restorations. Although in recent years there have been many new studies about seagrass conservation, seagrass was once considered as not important compared to other marine animals, and there are still a great number of people who would consider seagrass loss as a necessary and minor problem that is not worth funding. Considering the limitations of the results, there are a few suggestions that I had come up with. Most importantly, as mentioned earlier, it would be important for further trials to be conducted using the combined methods to check for any other elements that might interfere with the successful results. Once the transplant method can be conducted in areas, seas such as Belize or in the Southeast Asia that are habitats to many endangered species can be subjects of restoration projects. The Belize Sea in particular is a steady current and relatively shallow area which would fit all the conditions for the method. One last suggestion would be spreading awareness of the issue. One of the limitations of the research was that this research would not regulate the human activity that harms the seagrasses. However, by spreading awareness, it will likely change the behavior to reducing such harmful human activities. I would take the specific example of integrated planning done in Belize starting from 2017, which aims to safeguard ecosystems and balance multiple objectives in coastal Belize, which had prohibited coastal activities to an extent that it would not harm the seagrasses. Finally, I will be discussing the conclusions of my research. Based on the four main journals I had used as the foundation to my conclusions, I split them into two different categories, environmental and technological, as I had explained in my research questions. The results of controlling sedimentation levels and glacier species identity in the environment of the transplants will be separated into the environment category, which refers to the natural solutions in the thesis statement. Wire netting methods and controlling NaPH using the sample model given earlier would count as technological solutions. Overall, the results of this research may potentially benefit both the marine ecosystem and human life. The combined results show that the net wire method can come in very effective for rough tide areas and decreasing turbulence, while the sample model of NaPH controllers can help with overall growth. The interactions with other grazer species will solve the possible interferences with epiphytes, increasing the overall survival rates of these transplants. And the SSL feedback loop can also be considered in this process to regulate the light that the seagrasses will have during the recovery period, making the biomass growth rate more faster. As for the effects on marine ecosystems, the recovery of seagrasses will mean a home and food source for the organisms, thus increasing biodiversity and also helping with the endangered species. Effects on human life would also be large. Not only would the fisheries provide the fishing industry with a stable income, but it would also affect the environment. As mentioned before, seagrasses with other marine plants act as 45% of the photosynthesis that occurs on Earth. And as seagrasses have a high carbon sink capacity, restoration would have a big effect. Increasing the number of the declining seagrasses can accelerate the process and thus help prevent global climate change. Thank you for listening. These are the references that I had used.